We're on slide 19 of chapter 20. And we are going to talk about how animal viruses reproduce, sort of compare it with the bacteriophage life cycles. When the animal virus enters the host cell, a lot of times by endocytosis, it will then undergo a step called uncoating, and the outside portion of it will be released, and the DNA or RNA will be released from the outside covering. Now, sometimes budding occurs, and that is when the viral particles are released in a bud, and when that bud is going to have an envelope outside of it formed from the plasma membrane of the host cell. Okay, so retroviruses such as HIV contain an enzyme called reverse transcriptase. So there are many viruses that are retroviruses. HIV is one. The cold virus is actually a retrovirus. So what is reverse transcriptase? Well, you have to remember what transcription is. Transcription, in Bio 111, we learned that transcription is the first part of protein synthesis, and it occurs in the nucleus of our cells, and it's when our DNA codes for messenger RNA. So the reverse of that would be RNA being converted to DNA. So in HIV or any retrovirus, reverse transcriptase is an enzyme that carries out reverse transcription, where it takes the viral RNA and converts it to cDNA, and that cDNA I think stands for clonal, but it's just, we call this reverse transcription. And then that cDNA becomes integrated into the host DNA, and when the host DNA replicates, the cDNA replicates. So it allows the virus to remain latent for years. We don't call this the lysogenic cycle, but this is similar. Now, something will trigger that virus to become active. Maybe stress, it may be a sickness, but something will trigger that virus to become active. And now the viral DNA undergoes transcription and becomes messenger RNA, and then the messenger RNA finds a ribosome and produces the proteins that make the new virus components. So new viruses are produced that way. So this is how retroviruses work. I'll go ahead and there we go. Okay, retroviruses work by the attachment is when glycoprotein spikes in the envelope, because these are enveloped viruses. So glycoprotein spikes attach to a receptor on the host cell. And HIV's host cell is called a helper T cell, which is one of our white blood cells. It's a type of lymphocyte. So it specifically targets helper T cells of our immune system, and that's how it can shut down our immune system. So the attachment part is when the receptor of the host cell and the glycoprotein spike of the envelope of the HIV, they match up, they pair up, and that's the attachment. The entry is by endocytosis, and you can see how the virus is entering into the host cell. And then the next step is reverse transcription, where the enzyme reverse transcriptase, which was actually part of the virus, that little blue dot, reverse transcriptase converts the viral RNA to cDNA, which again I think stands for clonal DNA, but that cDNA enters into the nucleus of the host cell and integrates into the host DNA. And we now call it a provirus. And it can remain latent for even 10 years, maybe longer, but it can remain latent for a period of years, but something will trigger it to become active and enter the biosynthesis stage, where it will begin to undergo transcription. that DNA will be converted to messenger RNA. The messenger RNA will find a ribosome, and it will produce proteins that will go on to become the components of the virus. And then the maturation, those components are going to be put together, new virus is produced, and it will leave the host cell by, this is what exocytosis or budding looks like, okay? Now, so other particles besides viruses are viroids and prions, where viruses are best known for causing disease in plants and animals like herpes, HIV, cancer. And viral diseases cannot be treated with antibiotics. Antibiotics have no effect on viruses. But viroids are just naked strands of RNA. I mean, they don't have a capsid outside. They are just naked strands of RNA that can cause many crop diseases. Prions are proteins, and they are proteins with a contagious tertiary structure. You may remember that proteins have 4 levels of structure, and that tertiary structure is their final 3-dimensional shape. So prions cause TSEs, which are neurodegenerative diseases. That means that they cause the nervous tissue in the brain to degenerate over time. They're untreatable and they're fatal. Some of these diseases are mad cow disease, which can infect cows and humans. BSE, which stands for bovine spongiform encephalopathy. Now, going back to viruses for some reason, oh, okay, I see why, because it's going to show you how the flu virus works. So the flu virus has 2 types of spikes, an H spike and an N spike. And that's how we name the different strains of flu viruses, is by their H and N spikes. But the H spike, the purpose of the H spike is to allow the virus to bind to the host cell receptor, the receptor on the host cell membrane. And there are 16 different H spikes. So you would say like H1, H2, H16. And then the N spike attacks the host plasma membrane, which allows viruses that are already mature to exit the cell. And there are 9 different N spikes. So you basically name the flu virus by the type of H and N spikes. So like you could have H5N1, H15N9, and here's H7N9 and H5N1. H5N1, I believe, is the bird flu, which can infect humans and birds. And it came originally from birds. This shows you how a virus that only was infecting birds can mix with a virus that only was infecting humans and become a virus that can infect both. Okay? And the way that happens is humans have to be in very close contact with birds. So they have to encounter the bird virus in secretions. So let's say that you have people who work in chicken houses or turkey houses closely with a large population of chickens and turkeys, and the mucus and secretions that come from the mouth and the nasal passages of the birds can mix in with the humans, can get into the human nasal passages. And in that way, the components of the bird and the human virus can mix and create a combination of spikes that can infect both the birds and the humans. Now, you may have heard of emerging diseases. Emerging viruses or emerging diseases are either new or uncommon illnesses that are now common. So examples of emerging viruses are coronavirus, which COVID-19 is one, Zika, Middle East Respiratory Syndrome, or MERS. AIDS is the disease. The virus is HIV. Even though it's been around since the 1980s, we still consider it an emerging virus. West Nile, encephalitis, Ebola, avian flu. So what are the events that can cause the emergence of a viral disease? So first, a virus can extend its range. West Nile was transported to the United States. So it can extend its range from one country to another, from one continent to another, and also viruses can undergo genetic mutations, random genetic mutations. Some are more have higher mutation rates than others, like the cold virus, the influenza virus. These have high mutation rates. So the virus can extend its range, or a genetic mutation may occur. And this is just showing you viruses the Zika virus, which is spread by mosquitoes. And it's showing you areas that are more likely to have mosquitoes, which obviously is the South. And then emerging viruses and where they began all over the globe. You can look at some of the different examples. Now we're going to talk a little bit about coronavirus and COVID-19. A coronavirus was first recorded in humans in the 1950s, but SARS-CoV first appeared in China in 2002. And then, but we have not had any cases of that strain since 2004. Now, MERS-CoV first appeared in the country of Jordan in 2012. And then the one that we know, all of us have been affected by it, is SARS-CoV-2, which is the virus that causes COVID-19. That originated in 2019 in Wuhan, China. And it is a single-stranded animal RNA virus with high mutation rates. And it does have an envelope, which you can break down with soap and alcohol-based sanitizers. We were all educated on this, during the shelter-in-place phase. We were all told about all this. The glycoproteins that are in the membrane are S, M, and E. So this is an electron micrograph image of SARS-CoV-2. And it's got the E protein, the S, and then the M. And then it shows you the life cycle. Basically, what you have is attachment through endocytosis. You have the glycoprotein receptors attaching to the host cell, glycoprotein spikes attaching to the host cell receptors, endocytosis, viruses inside, and uncoating occurs. So now you have the viral RNA, which undergoes conversion to directly to the viral RNA. It's not like a retrovirus. So it just, since messenger RNA can undergo translation, it's like it already skipped transcription. Okay, so the viral RNA is used to make the S protein, M protein, E protein, and another one called an N protein. And 3 of those proteins enter the rough endoplasmic reticulum, and they're modified, and then all of those that are modified go to the Golgi. The N protein ends up going to the Golgi as well. So the S, M, and E protein go to the rough ER first, and then the Golgi, and then the N protein just goes straight to the Golgi. And then all of the viral components are assembled and they are released from the host cell, which is our respiratory cells. Now, we're going to move on to the prokaryotes, and we'll cover section 3 real quick. Prokaryotes are cells that don't have a nucleus. They include the domains bacteria and archaea, and they are unicellular. And then we have Louis Pasteur's experiments. Louis Pasteur showed that you can keep a broth sterile if you use a curved neck on the flask. So he did a couple of experiments. First thing he did is he took a flask that had a regular neck with a stopper in it, and he boiled a broth, like a nutrient broth. He boiled it to kill or sterilize the broth or kill the bacteria that were in there. Then he opened up the flask, exposed the flask to like outside of the building, to the air outside. And when he put the flask, the stopper back on and waited, 89% of his flask showed growth. And how you see growth in nutrient broth is it turns cloudy. So there were bacteria growing in it in 89% of the flask that he exposed to the air outside. He did the same experiment inside a building, and 32% showed growth of bacteria. So less than outside, but still, bacteria is getting in the broth. So he then used a curved neck. And when he exposed that broth to the air outside, 0% showed growth, or 100% of the flask had no growth. You could say it that way. And the reason is because the air enters the flask, but the bacteria settle at the bottom of this curve, and they can't go up the incline. So a curved flask will keep the broth in the flask sterile. A curved neck will keep the broth in the flask sterile. Okay, so prokaryote structure, they lack a nucleus, a membrane-bound nucleus. They do have DNA in a nucleoid region. They do have a cell wall. Some can have a flagella, but they definitely do have a cell wall. They do not have membranous organelles like mitochondria and endoplasmic reticulum. And sometimes, in addition to their single circular chromosome of DNA, they can have accessory rings of DNA called plasmids, sometimes. So a prokaryotic cell is going to have the cell envelope will include the plasma membrane, cell wall, and then outside that a structure called a glycocalyx, which can be like a slime layer or it can be like a capsule, more of a solid. But it's like an extra layer of protection, and it helps the prokaryote to attach to other prokaryotes and to its surface that it's growing on. The cytoplasm will contain a nucleoid region where the DNA is. It will contain ribosomes, and in cyanobacteria, these are photosynthetic bacteria, there will be thylakoids that can carry out photosynthesis. Some bacteria will have flagella for movement. Some will have a conjugation pilus for exchanging DNA with other bacteria. And then some will have fimbriae for helping them attach. And this is a picture of the structures that you might find in prokaryotic cells. They all have a plasma membrane and a cell wall and a capsule, which is called a glycocalyx. They all have ribosomes, and they have a nucleoid region that contains the bacterial chromosome. They all have cytoplasm. Some have flagella, some have conjugation pilus, some have fimbriae. And reproduction is asexual through binary fission, which means splitting in half. They can reproduce in as short as 12 minutes, but sometimes it's longer. They have high mutation rates. Prokaryotes are haploid. They only have a single circular chromosome. So a single chromosome means they're haploid. Diploid means 2 chromosomes. Some can form endospores. This is showing you a picture of binary fission. And lastly, there are 3 ways that bacteria can recombine their genetic material. Conjugation occurs when you have 2 bacteria. One is called the donor cell and one is called the recipient cell. And what they do is the donor cell passes DNA into the recipient cell through a conjugation pilus. So conjugation occurs through 2 live bacteria and a conjugation pilus. Then transformation occurs when the bacterial cell picks up pieces of DNA from other prokaryotes, which are either alive or dead in its environment. That is called transformation. So that's when a bacterial cell picks up pieces of DNA from other prokaryotes, not through a conjugation pilus, but just from the environment that it's growing in. And transduction is when DNA is transferred from a virus to a bacterial cell. And that virus, remember, is called a bacteriophage. So a bacteriophage it serves as a vector. In other words, it carries DNA from the bacteriophage virus to the bacterial cell. And we'll start with section 4 on bacteria. This is slide 43. We'll start with this one in the next recording.